Okay, so welcome back to the post-lunch session. And so the first talk of the session will be once again by Professor Maitri Narasimha, and she'll be talking about moving and shaping living structures. So thank you. Please, over to you.
Thank you very much. So very happy to be able to tell you a little bit about the kinds of things that interest us in the lab. So what I'll try and do today is move from where I left off yesterday to focus on shaping and moving not single cells but groups of cells. So a little bit about collectivity again, to continue on a theme that we've been hearing earlier today. But to try and tell you how tissues are sculpted and how organs reach their final destinations. And the point I want to make is that although we think of these as collective phenomena involving lots of cells, one of the things that I will highlight is that heterogeneity within a tissue with respect to what individual cells within a tissue actually do is essential to drive the shaping and movement of tissues. So before I begin, I would like to thank the people whose work I will keep talking, keep mentioning their names as we go along. So is there a pointer? Just the point. Yeah, thank you. So these are people currently in my lab. Some are missing from one photo, so there are multiple photos, and I will also highlight collaborations as I go along with Vijay Krishnamurthy at ICTS and Mandar Inamdar at IIT Bombay. Okay, so where I left off yesterday was to, one of the things I did mention yesterday was that the genetic basis of morphogenesis is a relatively new idea with respect to the effectors of morphogenesis. Mathematicians and physicists and computer scientists had ideas before biologists did. And these people here, Yanni Nusslein-Volhard, Eric Wieshaus, performed what is sort of perhaps the most interesting type of experiment in genetics, which is to randomly mutate the genome, right, with the hope that they will affect, those mutations will affect the functions of genes, and then try and isolate mutants in which a pattern, in this case the larval pattern with all its segments, of which Shashidhara spoke about a little bit, deviates from that of the wild type pattern. So what you see in this picture from the cover of their Nature paper in 1984 shows you what a wild type larva, and when I say a larva, I really mean a maggot of the same kind that lives inside mangoes and other bananas and other fruit, what the wild type pattern of a larval cuticle looks like. And through this sort of mutagenesis experiment, they showed for the first time that certain changes in the sequence of DNA would cause larvae to have patterns distinct from that of the wild type. And what you see here is that even if you just counted the number of bright bands in the larval cuticle, which are these dark bands that you see here, just visualized differently, you will see that some of these mutants actually lacked many of these bands. This alerted them to the fact that mutations can actually affect pattern. And through this type of experiment, they deduced that the genes in which those mutations were found were somehow important for the elaboration of this pattern. And this sort of experiment, which we call a genetic screen, actually allowed for the mutant bloom of the 80s. So they generated tons and tons of mutants and actually made them available to the community and said, if you want to work out how this gene regulates pattern, you can have it. And that really drove a lot of research into the genetic basis of morphogenesis. It was also the second contributor to why Drosophila became a very good multicellular organism model to understand not just morphogenesis, but a whole host of other things. So, and that led a lot of people to ask, how do genes regulate pattern? And so just a recap of what I kind of started off by telling you yesterday, which is that morphogenesis is the generation of form from simple beginnings to complex structures. If you focus on morphogenesis at the cellular scale, it's often accompanied by the regulation of cell numbers, either by cell division or cell death. It's accompanied by the regulation of cell types. This is a process biologists call differentiation, and that essentially means you go from having a stem cell that's capable of giving rise to many different types of cells to it getting somehow channelized or canalized on Waddington's landscape, so that some particular, sorry, I don't know why something happened, just give me a second. Yeah, to getting sort of canalized along a path to become a particular type of cell. But importantly, it's also accompanied by changes in cell shape and cell position, and I will just call this by morphodynamics. And it's all of this happens through the interplay between molecules. These molecules could be transcription factors that regulate the expression of several target genes. It could be through signaling. By that I mean operating at the level of proteins that work in pathways and amplify or ameliorate effects. It could be just through associations of proteins, through the formation of large protein complexes that affect functions. But it's also important to realize that mechanics plays an important role. And when I say mechanics, I really mean force generation through the link between the membrane and the cytoskeleton. And particular emphasis on molecules on the membrane, proteins on the membrane that fall into the category of adhesion molecules that I spoke about yesterday. So what force generation relies on? So if you have to deform a cell, right? If you have to change the shape of a cell, then the way you look at whether the shape has changed is to look at what the outer surface of the cell looks like. And in order for the outer surface of the cell to look different, any force generating mechanism that's inside the cell will have to be linked to the membrane, and key proteins that facilitate this are adhesion molecules. So what I'm going to do today then is take you through some features of how you shape a maggot, a fly maggot, with some key morphogenetic events that my lab focuses on. So this is a movie of a fly embryo that's already in which the zygote, the fertilized eggs, already divided a lot. So every dot that you see here is a single nucleus, therefore a single cell. And it's you can see it because we've tagged GFP to histone that's present in the nucleus. I didn't make this movie, it was made by Philip Keller at Genelia Farm, using a microscope that allows you to see the dorsal side, which is like the back of the embryo or larva, and the ventral side, which is the front of the larva, at the same time, right? And when I play this movie, what I want you to appreciate is how much of a change in shape of the whole embryo there is. And so now when I play this movie, you'll see that groups of cells are sort of, getting translocated. Groups of cells here are getting squished, only for the groups of cells that translocated here to sort of reverse gear and go backwards, so that this part here will end up eventually here, effectively unfolding the axis of the embryo. So it's almost as if the embryo was sort of bent so that the head and tail were at the same place, or at the same end of the animal, and that this reversal actually puts the tail end back at the tail and moves it away from the head. That's a process called germ band retraction I'll talk to you about. The other process, I just rewind this a little bit so that, so the other process after the tail's gone back, you'll see that this elliptical group of cells actually contracts, meaning that the area of the ellipse reduces over time. And in the process of doing so, it brings these two sort of flanks of the embryo close to each other to form a continuous epithelial sheet. So the ellipse actually goes inside the embryo, and the two ends or the two arcs of the ellipse actually form a straight seam in the middle. And in the way that that process occurs, it resembles the way wounds heal in the body. When if you make a cut on your skin, let's say, the two edges of the two free edges of the skin sort of gape to form arcs, but the healing process actually brings those two arcs to form a nice straight seam, right? So I've just shown you two of the morphogenetic processes, shape transformation events that my lab is interested in. And if you just watch these movies, you'll see that there's some sort of, I call it fluid morphogenesis because it almost feels like these cells and groups of cells are flowing. And in fact, if you take away the outer covering of these embryos and poke a needle through these embryos, fluid will, it's a, what you'll see is like some fluid mass, right? What this sort of morphogenesis must clearly involve is a fine balance between being deformable, i.e. to change shape so dramatically, but at the same time not just acquiring some random shape. I mean, there's clearly a shape, and embryo after embryo you'll see the same thing happening. What it also involves is an interplay between molecules and forces, and you'll see some groups of cells are pushing and compressing others, other groups of cells might be stretching some other groups of cells, and so on. So there's a lot of deformation that that processes could be influenced by. The third thing I want to emphasize is that sort of these processes require integration of information across multiple scales, and in particular from molecular scales to tissue and organismal scales. And these are the three broad questions that we've been trying to investigate in everything we do. And as I said, there are two processes that we've primarily focused on. One is the process where I showed you that this embryo that looks like it's folded upon itself so that the tail end is close to the head. This is the head of the maggot, this is the tail. Moves backwards so that the tail is at the tail end. That's the process. It's called germ band retraction. The other, where this elliptical tissue contracts and brings the two arcs from the right and left side close to each other to meet in the middle to establish continuity of a sheet that was otherwise, in which the flanks were otherwise held apart by this intervening elliptical tissue, is what we call dorsal closure. And these are the two things I'm going to talk about, so just emphasized here. And so I just want to say that all of the morphogenesis I'm going to focus on is what I call epithelial morphogenesis, because we're really looking at how a sheet of cells, which is a monolayer, single sheet of cells, deforms, right? We're just keeping it very, very simple because this is complicated enough. This is a physicist's eye view of an epithelial sheet with nicely packed hexagonal cylinders that have, you know, all these parameters, height, area, all of that stuff, apical tension, basal tension, etc. But in reality, there's almost no sheet in any animal that's actually this flat. Most sheets are curved, have curvature somewhere, and so it's not really possible to even pack cells like this in an epithelial sheet. And very recently, and this work made headlines even in the Hindu on the front page, that mathematicians had discovered a new cell shape, and this cell shape was called scutoid. And what it basically shows you is that cells on a curved sheet are packed in such a way that cells, the top end of these cells may have neighbors that are different from the bottom ends of these cells. So there's some sort of twist that these cells aren't sort of straight cylinders like that, but you know, they could have different neighbors apically and basally. And this was sort of offered as a packing solution on curved epithelial sheets. And so the shape that I'm actually talking about was something, this one here, scutoid, where you'll see that there are these additional faces that allow for different interactions at the top of the cell relative to the bottom of the cell. So we have to remember that epithelia in real life may not exactly be like this, but somewhat more like this. And so the things that I'm going to focus on, of course I'll tell you about those major morphogenetic movements, but what I want to try and tell you is also that one part of what we're interested in is how you build smaller multicellular patterns, right? One of them is what I call a seam, right? And this seam is the end result of this process here, where these two arcs actually meet in the middle to form a straight seam. If you zoom in on these seams a little bit later on, you'll actually see that they go from an arrangement where cells are sort of meeting head on like this to form plus or T-shaped junctions to becoming like this, which is more like how we arrange bricks when we build a house, with this staggering or interlocked configuration. So one thing we've been focusing on is how do you build seams like this? And we believe, like architects and other people believe, that this sort of seam likely has a greater mechanical stability than something where you just have two cells meeting head on like that. The other pattern we explore is the formation of a rosette. People like Madan would like to think of this as an aster-like configuration in the language of active gels, etc. But the idea is that you have a central cell that behaves or is shaped differently from its nearest neighbors. But this pattern is pretty local because it only involves this cell and its nearest neighbors, but does not involve its distant neighbors. So we call this a rosette because of its resemblance to a flower. The third is a placode, which I will come to soon after this introduction, which is a sort of radially organized structure where you can identify 3 distinct zones of cell shapes, a central one in blue, a sort of middle one, I'll show you better with cartoons of this, and a lateral one that are characterized by differences, let's say in the shape isotropy, with these central cells looking more or less isotropic, but as you go away from the center, cells become more anisotropic. So there's a sort of anisotropy gradient that looks a little bit like a cut onion, right? If you look at an onion that's cut in the right orientation, you will see that the central layer is a little bit thicker, and you get sort of thinner and thinner as you go along. So those are the structural patterns we're interested in, but as I said, we're also interested in mechanical properties, and in particular we're looking at deformations, flows, and here what you see is how a group of cells is actually, the tail end of the embryo that I was talking about makes its way back to where the real tail is. And we're interested in what forces contribute to these flows, right? We're also interested in aspects that relate to precision in this particular case of how you can take 2 curved arcs and make a straight line that that's perfect in the sense that there are no defects, right? In other words, I don't have bits that aren't fused and bits that are fused. From a genetics perspective, we're interested in understanding the network diversity of proteins that are present at the membrane cytoskeleton link, and we ask how is it differentially regulated to allow for different morphodynamic features. And we're also interested in the molecular basis for differences in the way the mechanical properties of cells differ at one end relative to the other. Okay, so I'll start with germ band retraction, which is moving the tail to its final destination. And here we've focused on asking how do you build that onion type structure, the placode, and how do you shape the flows that move this group of cells to the posterior end. Now this is an important process. It's an important process because in many organisms, as I said, the axis, the head to tail axis is bent during early embryogenesis, and it has to straighten out by the time the embryo is born. So this is the process in the fly that achieves that axis straightening. The orange colored tissue here is the same elliptical tissue that I was talking about that contracts later on after this process is complete. But what this process also does by way of towing is to move organs that are seated underneath the sheet to their final destination. So if you have to put the back end of your gut to the right place so that your gut functions in the right way, this is the same process that ensures that the back opening of your gut is actually at the back. I mean, not doing this would result in the back opening of your gut being somewhere in front, and at least humans will realize that this is not a nice thing to have, right? So, so the blue bits here are the gut, and this is what I was talking about, the hind end of the gut. It's actually not at the hind end. To begin with, the opening is at the top of the embryo, and the way it gets to the back is through this process. And so, but it also positions all of the segments in a linear order. So here the tail end segments were at the top, but by the end of this process, those tail end segments are back at the tail end. Okay, so we can actually watch this process happen at fairly high temporal and spatial resolution, and what you will see here are two colors. One in green marks the outlines of the cells. There's a haze of red here, ignore the haze of red, but the foci of red here mark the tail end of the embryo, right? And these large green cells are those orange colored cells that I showed you in the previous cartoon. And what you will see when I play this movie is that this sheet of large green cells is folded at this end, and as I play this movie, you will see that those folds vanish, and the sheet is now like flat. This part of the embryo, you will see that this red heart-shaped structure here moves all the way to the back. So I'll play that. So you'll sort of a little slow, but you will see that unfolding of this folded sheet is happening. So you can begin to see more and more rows of cells until somewhere at this point you'll see that you can see all those folds unfold and the red bit goes all the way to the back. So we think of germ band retraction as a sequence of unfolding events. I say sequence, but it's not necessarily one following the other, but two things happening sort of concomitantly, but both of them are straightening out. And the work that I'm going to present is work initiated by Shrudeepa Nandi here with Anchal Balse. Both of them have finished. It's sort of being taken over by Charvi Ravichandran, and we've collaborated with Vijay Krishnamurthy and Mandari Namdar on this work. So back from the screens of Nusslein Bolhard and Eric Wieshaus, we knew that there were mutations in genes in which this process failed to complete. And just like Professor Bose was talking about why genes got names like Van Gogh and Starry Night and all of that, these mutations or these genes were called hindsight because the hind end is actually close to the eye, or U-shaped because the straightening didn't happen, or tail up because the tail was on top, not at the back. And so we've known for a long time that there are genes which regulate this process, and we've also appreciated the importance of this axis straightening across organisms, although maybe the mechanisms are slightly different. In humans, you can have something called congenital kyphosis, which is of course due to a failure of segmentation, but in many cases you'll actually see that segmentation can be intact, but you will have adults who are sort of bent like this. You can often see them on those little wooden platforms with wheels moving around like that. But what I also want to use this to tell you is that the positioning of the internal organs, here again I'm talking about the opening of the gut at the back, very similar in flies and in humans, and that's why we sort of decided it was worth our while to figure out how this happens. From a fundamental point of view, we wanted to really understand how does an epithelial sheet unfold, because much work has gone into understanding how a sheet bends, and we know how sheets fold quite well, but what still remained unclear is how to unbend a folded sheet. And so that's why we sort of also decided to look at this, and we sort of really set out to ask how does this unfolding happen and how do cell fields become positioned, by simply looking at the kinetics of the process, the topology and geometry of the tissues, and the behaviors of cells that participate in this process. And so much of what I'm going to show you is what we've learned from these investigations. And so first, and I'm not going to show you the movie with the PIV arrows here, but from movies of this kind where individual nuclei are labeled, you can actually use particle imaging velocimetry methodologies to get a feel for the velocity of this large scale multi-collective movement. And what we saw from movies like this is there's a fairly long phase during which there's very little or no movement, followed by a phase when the velocity continues to increase up to a point, and then a phase where the velocity actually drops down. So what this tells you, there are two graphs here. One is superimposed on the other because I didn't have the time to sort of put them all on the same graph. But what this tells you basically is that the retraction or unfolding speed is temporally regulated. So the tissue has a mechanism by which it can regulate the speed of its movement. And again, I think that's a sort of fundamental question about what mechanisms does a tissue use to regulate how fast it moves, and I'll come to some indications we have about that. The other thing I wanted to point out that's interesting about this process is that cell fields within this tissue change shape and position. So even though I talk of this as a monolayered sheet, the first thing you'll be able to see, and here we've labeled myosin, be able to see, and myosin lies close to the plasma membrane in addition to other places, so it also allows you to get a sense of the shapes of cells. And what you will see when I play this movie is that groups of cells together change shape, but single cells also look heterogeneous in shape. This movie is high magnification, so the frame needs to be shifted each time. Once this red bit goes out of the frame, we bring it back to here. So that's why you'll keep seeing that the red bit always appears here and goes back and then comes back again. So I'll play this movie now, and what you'll see is this red bit, which is the hind end, is moving back. It hasn't gone all the way back, so we move the frame, bring it back here. But you'll start to see this onion ring-like pattern appear in the region that will become the placode. And then the red cells actually lose their proximity to these large cells. And there's a space here between this tissue here and the red cells on the surface. There are of course other red cells that are not part of the sheet that we won't worry about. But essentially what's happening is that this entity was close to or opposed to, I'll just play this back again to explain what I mean. So if I go back here, we started off with the red cells being close to these large green cells. You'll already begin to see here that you have a sort of layered, radially organized concentric layers appearing that are marked by supracellular organizations of myosin. And as I move, that layering becomes pronounced, but you'll see that this red structure here actually changes shape so that it becomes first elliptical and then pointed in the opposite direction. So in a way it's a bit like you're taking a heart that has its bumps placed near the head to making a heart that has its bumps placed at the hind end. So that's a major shape transformation. And then tissues that were once close to each other move farther away from each other. If you look at the same embryo in a lateral view, by that I mean instead of looking at it from on top, you're looking at it from the side, roughly at the middle of the embryo, you'll see this red haze that I was talking about. That's autofluorescence from the sheet that surrounds the embryo. But what you will, and these green cells are those large green cells. They initially overlap the red cells. These are the red cells that form that heart-shaped tissue. All I want you to notice here is that these cells are sitting on one substrate here, which is the margin of what looks like a black hole. And then another substrate here, which is the sheet or the covering of the embryo, which is really what keeps it together. When I play this movie, you will see that there are dynamic interactions. Cells interact with the substrate on top. It's a bit like if I have my hand on the ceiling, I don't have my foot on the ground. If I have my foot on the ground, I don't have my hand on the ceiling until this attachment with the top disappears. So what I just want to say is that this unfolding happens under increasing confinement. I've just indicated confinement like this to say that eventually all of this is happening between two substrates, and it's associated with dynamic substrate switching. Okay, so now if you look closely at the organization of cells within this heart-shaped entity at the tail end, what you will see is the progressive appearance of these concentric layers of cell shapes, with the central cells being sort of more isotropic in shape, and there's an increasing anisotropy as you go from, when I say center, I mean close to this midline, and on both sides of it this is true. As you go away from it, the cells appear compressed in one axis and stretched in the orthogonal axis, right? This is in the early stages. The late stages, I showed you that the shape of that entire domain first becomes elliptical and then makes a flip. So now you have a heart with its humps on the other side, and that's mediated by constriction of the cells at this broad end so that it becomes pointed, and in something that we've likened to a collective T1 transition. T1 transition is a word physicists here will know. Come to that again. In something that we've called a collective T1 transition, groups of cells exchange neighbors so that these big cells are no longer neighbors with these cells, and a field of cells comes and occupies the space in between them, thus sort of repositioning the red cells away from these large green cells. And if you look closely at how this happens, actually a group of cells that are closest to the midline make this constriction first and move backwards. So vertical columns of cells become now horizontally placed, and the more laterally placed cells constrict next, so that cells that were on the right and left sides away from the midline come and lie more anteriorly. So this is how we're moving not just this entire hind end of the embryo backwards, but also repositioning cell fields so that things that were close to this midline and organized vertically end up being horizontal and placed closer to the tail end than cells that were more laterally placed. So we spent some time trying to understand what was actually happening in this organization, which I should point out we got excited by because an old anatomical term, a placode, is perceived to be the beginnings of many organs. Many organs, if you read anatomy textbooks or embryology textbooks, there are so many different placodes. A placode that helps the formation of your ear, a placode that helps the formation of your eye, and so on. And we thought this looked quite a bit like a placode, and we said, you know, it'd be interesting to understand how you pattern a placode and see whether there might be something generic there. And so what we saw was that the cells in the center are not only smaller in area, but they're also more isotropic. By that I mean their aspect ratios are close to 1. But as you go away from the center to where these yellow lines are, you see that their aspect ratios are larger, but also increase up to a point before they reduce a little bit. But what was also interesting is that the central cells are, for a large part of this process, predominantly hexagonally packed, whereas the lateral cells, that includes both the middle cells and the outer cells, deviate from this hexagonal packing in that only about 30% of cells are hexagons. The majority of them have either far larger than 6 or far fewer than 6 sides. And this is used as a proxy for the propensity of cells to rearrange in a sheet, because cell rearrangement, exchange of neighbors within a sheet causes changes in sidedness because of cell loss or cell gain. And so typically if you think of a hexagonally packed epithelium, you would consider it stable. If you think of an epithelium that deviates from hexagonal packing, you sort of think of it as a more dynamic epithelium. And the sorts of mechanisms in place to cause cell rearrangements are what we call T1 transitions, where if you just focus on this region here, where you have a group of 4 cells, right, labeled in different colors here, you will see that this contact between the purple and orange cell shrinks to a point to create four-way vertex. But this vertex resolves in the opposite direction to push away the red cell, the orange cell and the purple cell, which cease to be neighbors. The blue and yellow cells, which were not previously neighbors, now become neighbors. So this sort of rearrangement is typically also accompanied by the change in the sidedness, and therefore what this sort of hinted to us was that maybe as you go out, away from the center, cells have a greater propensity to exchange neighbors. The other thing people think about is that the more rearrangeable an epithelium is, or the more likely the higher the frequency of these T1 transitions, the more fluid, I put that within quotes, the more fluid that part of the epithelium is. And the more hexagonally packed they are, the more, or the less fluid or more rigid that group of cells is. So one of the things that we thought about was that maybe in addition to these shape anisotropy gradients that characterize the placode, there must also be a sort of difference in the relative fluidities of these zones of cells. And so with Mandar and Vijay, we found ways to work out whether the mechanical properties, and in particular the magnitude of pure shear, might be different as you go from the center outwards. And what that allowed us to see is that for a long distance, so between the central and middle zones, there's sort of no real change in the magnitude of pure shear. But the moment you sort of hit this region here in the periphery, the magnitude of pure shear, although small, still goes up significantly compared to the more central region. So what that led us to think is that there's probably also a mechanical difference between these 3 zones. Um, the third difference that's also interesting, and I should just point out, a placode in anatomical terms. When I say anatomy, it's sort of neglecting cell biology, but just looking at whole structures. A placode is conventionally defined as an epithelial thickening, right? A thickening. And so we were sort of wondering, what is this thickening about? And so one of the things we decided to look carefully at is what it looked like in cross section, right? And there are two things I want to point out here. So I think the CML, okay, so the CML is here. So the central cells are close to this midline, the middle cells are here, and the lateral cells are there, schematized here. What you'll see is this placode is actually a structure that is characterized by differences in height in the two zones. Till now I was telling you about cross-sectional areas mostly and aspect ratios mostly from this end. But what you will see is that it's a little bit like an inverted dome with a sort of summit here, but this part of the placode also forms a bit of a dome in the sense that the cells that don't form the placode have a different radius of curvature. So we're actually thinking about a structure that has a curvature that's distinct from the non-placodal region, but also has an increased curvature on the inner side. So it's like a dome, it's like a thickening with two domes, one that's flatter than the other, right? And so really our interest has also been to understand how you pattern cells to acquire these three-dimensional shapes. And yeah, so I'll skip this and just get to, so that led us to ask, how do you pattern these distinct shapes? And clearly if, one thought we had was that the cytoskeleton somehow formed boundaries between these two zones, so that some cells were being confined by cables more strongly than others. And consistent with this, if you just look at the actin cytoskeleton, forget all these interesting other things just now, but if you just look at the cytoskeleton here, you will see a very nice sort of set of supracellular actin cables. I say supracellular because these are oriented and aligned not in a planar polarized way, like you heard this, in a planar polarized way, but in a sort of different kind of planar polarized way, in that the stretched circumferential boundaries of the cells appear to be enriched with actin, but the sort of compressed, radially oriented boundaries of these cells don't have those enrichments.
Yes, still looking at the germ band, right? So this is mark the actin in the germ band.
Mark the actin in the germ line.
Germ band.
Huh? In the, sorry, say again, I said I'm a little hard of hearing. Just one second.
Ma'am, just to clarify, we're still looking at the, so myosin SQR GFP has marked the actin cytoskeleton in the germ band, right? So that is still the retracting germ band.
So it's actually, so myosin is the motor that works with actin. So we're using it as a proxy for actin. And it's actually it's a GFP tagged protein that is driven with its own promoter. So it's actually there everywhere that myosin is. And myosin is an essential protein, virtually every cell. So what it actually does is, it's present in every cell in the embryo. We are just looking at the germ band. We are sort of focusing on the germ band. So what I was pointing out is that you see these planar polarized supracellular cables that are sometimes spaced closely, and in the central region here, you don't actually, I mean, you see the first cable around the central region, but the central cells themselves seem to have anisotropic myosin on some interfaces, but not others. But it's sort of randomized. But as you move outside, you begin to see this layered organization. So, so some still images of myosin are seen here. So this is sort of how we think about it, that in the central zone you have random enrichments of myosin, but as you move especially towards the lateral zones, you have these supracellular cables. And so where you have long cells, you also, cells that are circumferentially long, you also have circumferential actin cables, but those actin cables actually skip some cell rows in ways that actually tightly demarcate the zones that we've identified. So it looks like these cables actually form fences around the zones that differ in their morphodynamic properties, and just like the cell shape anisotropy, where I showed you that the aspect ratios of these cells, just go back to it. Sorry. Yeah, maybe I didn't show you this. So if if you look at the strength of anisotropy, it increases as you go from the center to the lateral region. And likewise, if you measure the anisotropy strength of the cytoskeleton, here we have both halves. This is the midline, and this is going away from the midline. So you can actually see that it goes up as you go laterally. So I am not going to talk about this part just now, but what that allowed us to say was that the anisotropy in the cytoskeleton mimicked the anisotropy in cell shape, and so we set out to ask whether the cytoskeleton actually governs these morphodynamic differences we see. And so what we did was to use genetic methods to perturb just this part of the germ band, the tail end of the germ band, with genetic ways of perturbing contractility, meaning perturbing actomyosin function. And what we ended up seeing, so these are two ways of down-regulating contractility due to the actomyosin cytoskeleton. If you look at the cellular organization here in this particular case, you'll see that you see some features of radial organization. You might see that these streams are in fact a little bit curved, but in large measure you will see that the gradients are not so stark, in the sense that when I say gradient, I don't mean gradient in the true sense of the word, but you can't, these are controls here, by the way, right, unperturbed. So you see central cells, middle cells, lateral cells, increasing anisotropy. Here you don't see such clear differences, and in fact if you make some of these measurements here, what you end up seeing is that the percentage, okay, so this is about sidedness here, percentage of cells that are six-sided or deviate from six-sidedness. This was the graph I showed you before. You see that the central cells also now deviate from being predominantly hexagonally packed, and the lateral cells show a greater predominance of non-hexagonal cells. So it looks like, in addition to reducing the shape anisotropy, what you've also done is increase the propensity of cells to rearrange when you affect contractility. We can also use lasers to cut some of these cables, and in this experiment the orange side has a cut cable, the green side does not have a cut cable, and you will see that the edges of the cut, I mean where the cable was cut, is jagged, so it doesn't have that smooth ring-shaped morphology with sort of aligned sides. Oops, sorry. So we think that those cables, not pressing something. Yeah, the cables not only help prevent mixing between cells, but they also form very clear boundaries. But what's interesting is that with time, a new cable forms in the same place, almost as if the cells had a memory of their previous state. They form a cable there. The moment that cable forms, you see some smoothness, at least in the region where the cable forms. So a jagged edge becomes smooth the moment a new cable forms in the same place. So what this allows us to speculate is that these actin cables, they're not actually contractile, meaning that, I mean, we've seen that in the sense, I don't mean contractile, they're not shortening, right? But we know that they're tensile, in that if you take a laser and cut it, the two edges will recoil, right? So they're tensile actin cables that are not shortening, whose function is to act as fences, one to prevent mixing between the different zones, but also more generally within each zone to regulate the extent of cellular rearrangement. So one way you could think of it then is that these actin cables are somehow modulating the relative fluidity of the individual zones and preventing mixing between these individual zones. But what was sort of surprising to us was that if you affected contractility in this region here, the effect it had on the kinetics of retraction was to actually hasten the process. So the duration of the time it took for this end to reach a particular point, so we've divided it into phases, but the duration of this retraction phase was actually shorter compared to control, which sort of told us that this kind of organization acts a little bit like a brake or a dampener that prevents this tissue from sort of moving backwards at too high a speed, and we're still sort of trying to work out how this organization might actually act as a dampener. So in contrast, and I've not spoken about this, okay, maybe I'll sort of leave that. Okay, so the consequence of either perturbing contractility or perturbing just the actomyosin cable is that the tissue doesn't sort of get positioned to the same place finally. And what you'll see here is a lateral view of the embryo. This is the hind end, right? That's actually the anus of the fly larva. And what you will see when you perturb this is that it's spread over a large region of the back end, and it's sort of both so, and if this is a glorified view of the hind end of the embryo, like nobody's seen it before, because not that easy to image the hind end of the embryo. But I just want to point out here, these look like two masks, two faces, but I think it's easy for you to see that the shapes of cells and their relative positions are very different in an unperturbed embryo relative to one where contractility has been disrupted. So just using this as an end stage effect to say that even just perturbing a supracellular cable can have fairly drastic effects on the final positions of cell fields within the embryo. And this sort of just summarizes the kinds of things that we've identified. I didn't talk about this second part of retraction that allows the orange cells to no longer be neighbors with the green cells, but that's also something we've looked at. And essentially and here also there's a cable of actin that constricts to move those cell fields. I showed you that in the beginning. But in both processes, we have T1 transition-like events operating between cell quartets, here as I showed you, or between multicellular cell fields as I showed you here, and contractility disrupts both of these processes, and through it alters the way in which cell fields become positioned. So one part of what we're interested in is to look at why one of these cables constricts and if perturbed hinders the final phase of retraction, why the other cable does not constrict but seem to somehow act as a dampener or a brake of retraction. What positions them, what makes them different, these are questions we're trying to ask. I'll just, how much time do I have? It's 3 o'clock, past 3. Huh? Okay, so I should finish now, right? Oh. Okay, plenty of time. Okay, so fine. So I'll just take you through the other part that I was talking about. I mean, much of this work is relatively new, but I just wanted to say, as I said before, that all of these events happen under increasing confinement and with dynamic substrate switching. So one of the things we've been asking is, how does interaction of these tissues with substrates actually influence morphodynamics and kinetics? And so as a first part to that question, we looked at the molecules I spoke about yesterday. These are transmembrane proteins, integrins. They're adhesion molecules that allow cells to link to the extracellular matrix, and they bind ligands that are actually associated with the matrix. One of them is laminin. If you look at where integrins and laminin are enriched, you will see it's in the two halves of the onion that I've been talking about. So these are the dark spots that you see here that are exactly at the place where you see these concentric organization that characterizes the plaque wood. And if you look at where, I mean, if I cut through this embryo like that to get a cross section, you will see that a part of these cells, right? And so typically the central and middle cells sit on a substrate that's enriched in both integrins and laminins, but these shorter lateral cells appear not to be sitting on a substrate that is enriched in either integrins or laminins. So this tells us that there is some heterogeneity with respect to the substrate on which these different zones sit. This is sort of cartoon of the same thing, except if I make a cut like this, in which case the central zones are sort of in the middle with some mirror image symmetry. I mean, in principle, there should be central, middle, lateral, and you'll see that the central and middle zones are sitting on a substrate that's enriched with integrins and laminin, but the lateral cells are not. So there's heterogeneity with respect to the substrate on which they're sitting. And this was interesting to us. I mean, many people who work with cells in culture and are interested in mechanics typically grow cells on islands of substrate of different shapes, different sizes, etc., and look at whether what kind of patterns you see when you alter substrate stiffness, when you alter substrate composition, and so on. So for us, this looked like a natural place where substrate heterogeneities and their role could be tested. So where we are at now is to sort of really start disrupting these interactions to ask how does this affect morphodynamic and mechanical properties. And to do that, we use again a genetic tool that allow us to disrupt these interactions. And if you use this tool in the wing, which you heard about this morning, you create a blister in the wing. Here you see a collapsed blister, where the blister arises because this wing is actually a bilayer, a dorsal and a ventral layer that are held together by integrin extracellular matrix interactions. So if you mess around with those interactions, the wing has a blister. So we use the same tool, which is the overexpression of a kinase that negatively regulates these interactions, to ask what happens to the organization and dynamics of this process. And I'm just going to show you here that if you just look at this cellular organization that I've been talking to you about all this while, where you have central, middle, and lateral cells in a unperturbed embryo, right, versus one where you've perturbed it, you see several differences. One, that it's harder, especially on this side, to see any sort of radially organized structure. In fact, cell columns are linearly arranged rather than circumferentially or radially arranged. The other thing you'll see is that the cell shapes are not patterned in the same way as they are in an unperturbed embryo.
Excuse me, I have a question. So all these perturbations, they are done locally, right? It's not.
Yeah,
it's not
they're done
repetition.
just to this part, this heart-shaped structure that I showed you.
Just curious, how
when
do
I
you?
say locally,
Right.
I mean it's a genetic tool where you can induce a particular perturbation in a spatially and temporally regulated manner. So here, in the fly, it's called the US GAL4 system, where you use. So this GAL4 is expressed only within this heart-shaped structure, nowhere else, right? And then, so that any perturbative transgene that can associate with GAL4 will also induce a perturbation only in that region. So it's just the future hind end of the embryo that's been perturbed, right? And the consequence of that is that you alter quite drastically the organization of the cells in two ways. As I said, you don't see a striking radial organization. Cells appear to be more in linear columns. You also see that the central cells are far larger than what you see in controls. And some of these cells are also anisotropic in ways that the central cells are normally not. So again, you can look at the three zones of cells in controls and perturbed embryos and see that where in the control some gradients, like for example the anisotropy strength shown here in aspect ratio increases as you go from central to lateral. In the perturbed embryos shown here in this light pink, that gradient is flat. All cells appear to, all zones of cells have roughly the same aspect ratio. So one of the things that we think is happening is really that, yeah, so again, if you look at the proportion of cells with 6 sites, I told you that in control, central cells had the highest proportion of cells that were hexagons, and the lateral cells had the lowest proportion. When you perturb integrin ECM interactions, you know, there's a flattening of that difference. So in a way, what this is doing is to sort of homogenize the population so that the 3 zones are not any longer markedly different from each other. What it does do is make these central cells much larger than they used to be in control. So they're making cells larger, but they're taking away these zonal differences in morphodynamics. And some of these features recapitulate what we see when we perturb contractility. The other thing that we see, and for some reason these movies are not playing, but these are again PIV analysis of controls and mutants, and looking at the velocity profiles of controls and mutants. And the point that I want to make here is that while control embryos show this rise and then fall, like I showed you before, what you see in the mutants is that the velocity does appear to rise, but after a while there's some sort of instability. By that I mean that the velocity drops, rises again, drops, rises again. And so one way we're thinking about it is that perhaps rather than regulated sliding or movement or regulated unfolding, at some point there's some sort of something that looks like a slippage, if you will, that probably accounts for these rises and falls of fairly high amplitude that we're seeing in these mutants, and that's something that we want to look at a little more carefully. So we're also looking at other ways of affecting integrin ECM interactions. Some of them look like what I just showed you. Others, where you try and degrade the matrix, look a little bit different, and we're still sort of trying to work out what accounts for these differences. So broadly, where we are with germ band retraction is at a point where we want to look at what properties of these interactions might account for the loss of morphodynamic patterning and its effects on tissue flows. And in particular, we're interested in looking at the impact of the composition of the substrate, its topology. By that I mean, how is this matrix actually organized in space, and stiffness, which can be also due to the substrate, the material properties of the tissues on which these cells sit. And I told you that they switch substrates while this retraction is happening. So it doesn't necessarily have to be about composition as much as how stiff is this substrate on top, how stiff is this substrate below, and these are things that we're sort of trying to have some measurements of. I'll just quickly tell you by way of what other things we do in the lab. In this process that resembles wound healing, dorsal closure process, we're interested in understanding how you actually build a seam. As I said, if you look at this seam that's formed by the leading or the leader cells in these two flanks, you see that the way they make contacts changes over time, evolving to this interlocked configuration. But what we also want to know is how the packing changes as these cells approach each other to make contacts, and in particular if shapes become more regular in 3 dimensions as you come closer to meeting and forming these new junctions that you can sort of see here in the fluorescent signals that appear just as these cells come and make contact with each other. And so just one evidence that in fact these cells are more irregular is that if you look prior to the fusion of those 2 flanks, if you look at the cell shapes, you see as you go from the apical end to the basal end, you see that the shapes are different, even the neighborhoods are different. So the same cells are no longer neighbors. I mean, the cells that are neighbors here are not neighbors here. But after you've completed fusion, not only the shapes of the cells, but also their neighborhood is a little more stable. So you don't see as much difference between the top end and the bottom end of the cells, both with respect to its shape and its neighborhood. So in a way, what we're trying to get a handle at is when you build a seam and you want to make it strong and stable, what sorts of changes account for this stability, both from the point of view of shapes, but also from the point of view of the nature of the contacts which have been spoken about. Other thing we look at is how you pattern shape oscillations which happen in that elliptical tissue that I showed you, where if you watch this movie closely, there'll come a point when these oscillations sort of reduce, right? You can see it already reducing now, and then they oscillate less but contract more. So you can see it's sort of slowed down with respect to oscillations now. And so if I had to put a graph of how the area changes over time, you'll see that you have these shape oscillations that dampen, resulting in a net reduction in area. The sorts of things we've been looking at is how do you regulate these transitions, the extent to which the membrane forms. And what we've shown is that this is governed by the interplay between actin and microtubule cytoskeleton dynamics. And this is something we're looking at. We've shown that myosin is primarily responsible for these oscillations, and it goes from myosin that's not cortical, goes from small seeds that coalesce to form a blob that then moves and then disaggregates and disassembles. And this cycle is actually dependent on microtubule organization around the myosin that forms initially a cage-like configuration and then a tail that we think respectively helps myosin coalesce and then move. And we're still working out some details of this. Perturbing microtubule dynamics actually also causes instabilities in cell shape, with cells dramatically collapsing and then expanding again, rather than this patterned reduction in cell area. The third thing we're looking at is how you build these rosette-like structures. And what you'll see here is that this rosette emerges, this pattern emerges through the constriction of this central cell and shape transformations in its nearest neighbors that have a long axis perpendicular to their interface with this central cell. We've shown a long time ago, and in fact this is my first student, Meghna, we've shown a long time ago that this pattern actually relies on the alignment of microtubules in the neighboring cells along the same axis, and that one of the functions of this alignment is to also target flows of myosin towards this interface. So whether you look at cellular organization or cytoskeletal organization, it has this aster-like element to it. And with Madan many years ago, we suggested that this aster-like configuration that emerges must result from a tilt in the balance of forces operating on the interfaces. The cell with a net force now acting inwards as opposed to there being a force balance along those interfaces. And we likened this this process in which this cell actually constricts and delaminates from the sheet to sort of yield stress in materials where, you know, the behavior from elastic to plastic might account for the exit of the cell from the sheet. And one last slide to sort of say that a lot of the work we did with Madan actually involved our ability to trigger this delamination using lasers. And that actually allowed us, as you will see here, allowed us to actually capture these cytoskeletal rearrangements in the neighboring cells. So these are the neighboring cells here. This is the cell, you can see it, you can see flows of myosin that enrich here. And so what this allowed us to do from the sort of recoil or the expansion, the changes in increase in area that accompanies the release of pre-stress, we were able to sort of get some magnitude for the pre-stress and then argue that when the pre-stress exceeds or is equal to the yield stress, that's when cells might delaminate. And last but not least, we're looking at how interface deformability might be regulated. And in this particular case, we reason that these interfaces here must behave differently from these interfaces here. And so what we're trying to ask, those interfaces do differ with respect to bond length. So these red interfaces shorten, these green interfaces, which are these ones here between nearest neighbors, lengthen. The ones that are far away don't change much. And using, again, genetics where perturbations induce delamination, we were also able to identify different interfaces between, for example, green cells and non-green cells, between two green cells and between two non-green cells, and show that the composition as well, the length as well as the composition of these three types of interfaces is different. I think I'll stop there and say that I'll just use one slide to say that the process that I described before delamination also happens in many places in the embryo. And in fact, it's the way the nervous system forms in the fly, except that the way in which this delamination happens does not, this cell here is delaminating, but you'll see that the kind of rosette that forms when cells in the nervous system delaminate do not actually look as symmetric as the rosettes you see in the amniocerosis. So if I have to just show you a schematic, these rosettes look a little bit like that. And one thing you can see is that the number of neighbors changes during this process. So these rosettes are asymmetric and there's neighbor loss. And so I think what we're trying to ask is what governs these differences. And in particular, we're interested in asking whether the global morphodynamic state of the tissue might itself influence the local configurations of cells to achieve the same. I'll stop here and just end with a picture of past members in my lab, because I may not have stated their names but contributed to this work, and thank TIFR for funding us.
Any questions?
Hi, I have a couple of questions. So, are you saying that these rosette structures are more likely to see when you have delamination? The rosette structures, they're more likely to appear whenever there's delamination, or?
Yeah, so we've looked at it in the context of delamination.
I
You
see.
could say, in a way, when cells are hexagonally packed, you know, each cell has 6 neighbors. In some ways, that's, you know, what we mean when we say a rosette is that you have a center, and then you have a cell and you have its nearest neighbors, and you pattern the behavior or the morphodynamics of the cell in the center is different from the morphodynamics of its neighbors. And typically a rosette is something where a ring of petals, I mean flowers are mostly symmetric, if not along the radial axis, they're symmetric about some other axis. So when we say a rosette, we mean some sort of radially patterned and symmetric organization. We have looked at it in the context of cell delamination, right? In which the cell in the center is constricting, its nearest neighbors are changing shape, but their aspect ratios are sort of directed along the 2 axes in such a way that the long axis is always perpendicular to its interface with the delaminating cell. So I'd say that we started with delamination in the first tissue that I showed you, where we see nice symmetric rosettes. 2 things, symmetry is 2 things. One, that the cell in the center constricts isotropically, and the neighboring cells are sort of symmetric with respect to shape changes, and no neighbors are lost or gained during the process. But in another context of delamination, which I just used one slide to tell you about, we see that the constriction of the cell isn't isotropic either, and the rosette does not look like a sort of nicely radially patterned entity. When I say it's asymmetric, I'd say that you wouldn't really be able to fit a circle around the boundaries of the nearest neighbor. It's some jagged boundaries, different petals or nearest neighbors of different sizes, and the number of petals changes over time. Yeah. Does that answer your question?
Yeah. I have one more question, one last
so
question.
the point, what we're trying to do with our physics collaborators is to try and put different boundary conditions of contractility and adhesion to see if we get symmetric versus asymmetric rosettes, so to speak. Yeah.
No, thanks. I have one more question. So you mentioned that this play code structures, the play codal structures, they're very often primitive structures to an organ. But here the placoidal structure is occurring in the tail end, I suppose, right?
No, so when I say, no, so what I'm saying is that they're present in the embryo. So in the human embryo, for example, or in vertebrate embryos, before you can actually see an ear form or an eye form, you see that the region where the eye will form or the ear will form has a placoid-like organization. In the human, it takes quite a long time before that placoidal organization culminates in a visible eye. In our case, we're talking about a placoid-like organization being evident, let's say 6 or 7 hours before an invagination is made. For example, as a hind end.
Right. But in this case, to which organ does it? What organ develops from that
Yeah. So
tail?
it is really there to get the the back opening of the gut. So in other words, it helps get the outlet on the outside. But the outlet is not present till just as the embryo is ready to hatch. Right? So the larva has it. So if we put colored dye into some of these embryos in which position of that structure is affected by disrupting placoidal organization, our anticipation is that the colored dye will actually not come out. It'll get stuck somewhere. So some children are born with congenital defects in the gastrointestinal tract, which and typically a thing that medics do is to give them barium to swallow and then do x-rays. What that tells you is where the gut ends or where a block might be. Our thinking is that along similar lines, this placoid allows the hind end of the gut to actually sort of break open to the outside through regulated invagination, but that invagination does not happen at the stages that I showed you, but much later, right? After this thing goes all the way to the back. And our thinking is that that organization actually regulates the speed of this movement. Perhaps because you want to ensure that something's positioned right before you actually create the opening. Maybe it works like a checkpoint for positioning. That's a bit of a stretch, but yeah.
Yeah, so in the context of this dorsal closure, you were showing these oscillations. I was not really clear as to what, so I saw that there is these myosin clusters that you showed that move to the interior and then to the outside. Is that, and that is associated with some microtubule organization as well. So how does that lead to this oscillatory behavior?
How does it?
Lead to the oscillatory behavior?
How does it lead to the oscillatory behavior? I'll say details we're still working out, but one of the things we know now is that some of this is published. So the plus ends of the microtubule, which is the growing end of the microtubule, bind ROGF2, which activates Rho. We think that what it is likely doing is, and the microtubules actually organize with their plus ends around the myosin blob. I'll say this with a little, I'll say we don't have very high resolution images, but we do have high resolution images, but I'll say that the plus ends are closer to myosin than the minus ends. There could still be some anti-parallel organization. But we think the plus ends actually create a zone of ROGF2 around the myosin blob and actually allow its coalescence by activating contractility. But we don't actually have, I mean, we've shown that ROGF2 associates with the plus end, but we don't actually have a clear sense of whether ROGF2, whether microtubule depolymerization is necessary to release ROGF2 and then ROGF2 is just out there locally but unassociated with microtubule, or whether indeed microtubule growth of this against or around this myosin blob actually somehow pushes the blob through microtubule generated forces. That's something we don't have evidence for.
But is there actin enrichment also?
A little bit like an actin comet propelling a bacteria inside a cell. So microtubule polymerization against a barrier could move it, but we don't have evidence for that. What we're trying to do is to ask, I mean, we know that the microtubules are not closely associated with myosin as the myosin blob fragments.
Mhm.
But the details of exactly how they're working, we don't know. But what I'm quite excited about is that if you look very carefully, microtubules form a cage around myosin in the beginning, but then that cage is no longer, so there's an asymmetry. By that I mean it looks more like a crescent rather than a ring around myosin. And we always see that the plus ends and the foci of microtubule nucleators sort of form linear trails behind the myosin, which suggests that myosin is like this and the microtubules are sort of forming like a comet tail behind it with the plus ends close to the myosin.
But so as the myosin cluster moves around, you're saying that the microtubule network is moving along with that? As the myosin cluster moves, you're saying that the microtubule network or the cage, whatever, is moving along with the myosin cluster?
Yeah, so we can follow the tail for about 14 seconds behind the myosin.
Okay.
And then we lose it. When I say we lose it, either that means that microtubules are depolymerizing at some point, so we can't track them anymore. But that's not too far away in time from when the myosin also fragments. So our big questions are, one, whether myosin is influencing microtubule organization, and two, in what way is microtubule organization moving myosin. And visually that's been a little hard to crack, although genetically perturbing one, so perturbing either microtubule growth or microtubule nucleation, which happens sort of on the go through these foci of non-centrosomal nucleators, affects the amplitude, the pulse amplitude. Cells aren't pulsing. And in fact, what it appears to do is to drive them precautiously into that phase of unpulsed constriction. So they in fact constrict more rapidly, but they don't pulse. So we think that that microtubule dynamics is in fact governing where subcellularly in that apicomedial space myosin is going.
Okay, so if there are any further questions, we can continue them over tea. But for now, let's thank the speaker for a really interesting talk. So we'll break for tea. It should be outside, and we will